Okay, thank you very much. I really appreciate the opportunity to come here to present our latest method, cryo-to-struct, for building protein structures from Cryo-EM density map. So as we all experience, AlphaFold is revolutionized structure biology. So many people think AlphaFold now can predict the monomer structures very well, structures of small complex very accurately. Then they think the protein structure prediction problem has been fully solved. Actually, that is not the case. AlphaFold still cannot accurately predict the structures of large protein complex, large protein assemblies, and molecular machines. So the key technology that can determine such a structure is Cryo-EM. So Cryo-EM is critical for determining such structures for training next generation AI methods to predict the structures of these large molecular machines. As Janet pointed out yesterday, I think the Cryo-EM is the pillar of the next generation structure biology. Now let's see how the protein structure data has Cryo-EM determined protein structure data has grown over years. So here you can see this purple bar represents the number of the Cryo-EM density maps grow in the ISMB. They grow very quickly. The green bars represent the number of the protein structures built from these Cryo-EM density maps, also in the PDB that grows very quickly. Right now we have about 36,000 Cryo-EM density maps and about 21,000 protein structures, large protein structures built from these density maps. Another key thing is that the resolution of this density map has drastically improved. You see, in early days, most density maps have low resolution, less than higher than 15 angstroms, very few high resolution ones. Now you can see the density maps become much more accurate than before. Now, about 75% of the total density maps have resolution less than 4 angstrom. Now, the key challenge is how can we build a structure from these density maps? Now, let me give you a quick overview how Cryo-EM is used to determine protein structures. Then we figure out where we as bioinformaticians can make contributions to this critical technology. The first is that given the protein samples contain many proteins of the same protein, many copies. We first spread around a grid. Each well contains one protein, then freeze it, then use electron microscope to project the light through it, generate the images of many proteins or particles in those wells, and in the different orientation. So you generate many of these images contain the 2D shapes of proteins in all different places. Then from there, we identify those protein particles. Then from those particles, we build a 3D density maps describing the electron densities of atoms in the 3D space. That is indication of where the atom is and what type of atom it is. From those density maps, we can build the final 3D atomic protein structure. So that's called structure modeling. That's the problem I'm going to talk about today. There are already some methods have been used to build the density maps, protein structures from density map. The most widely used one is called a template-based approach. So I first get a structure for each unit, each chain of the protein complex from AlphaFold or template-based modeling. I map those chains into 3D density map. Then I try to refine it, optimize it to build a better structure. That's widely used method. Another approach is called the Novo approach. That is, I only use density map as input, nothing else. I try to build a protein structure. How well can we do that? Of course, you also can combine the Novo approach with predicted structures together, try to build a better structures. So today I'm going to talk about the Novo approach. Why we needed the Novo approach? First is practical need. So if I don't have the good structures of the chains, I have to build from scratch using ab initio approach. Another reason is that this Novo approach can be fully automated. Right now, the template-based approach needs a lot of human intervention, very laborious. It's very hard to reproduce. But this Novo approach can be fully automated by machine learning. Another thing is scientifically interesting because we really want to know what kind of structure information we can just build from density values alone, right? So that's interesting for us. And also it's timely because now, as I show you, there are a lot of density maps, a lot of structures have been built from density map. Really, it's time to build AI methods to automate this process. And also, surprisingly, it's very underdeveloped by the university field. It's so important, but a few bioinformaticians are working on this. We just started working on this 7 years ago. Now, so here's our approach for the lower reconstruction of the protein structures from density map. It has 2 steps. First is I want to use machine learning to identify atoms and their amino acid types they belong to. That's first step. Second, once I identify them, I want to trace those atoms together to build the protein chains. And we develop a AI methods to do this. The first is that give the density map as input. We break those density map into smaller cubics, just like words in a sentence. Then these are used input for a 3D transformer, pretty much like a ChatGPT for natural language processing, to generate the embeddings. Then finally, we use these embeddings to predict whether or not a vocal cell in the density map contain an atom or not, and what is the type of amino acid of that atom. So because some atoms may close each other, actually work so close to each other, will be classified as atoms. They actually belong to the same atom. So we need to classify them to build a identify all the atoms. Now these atoms can be used to build a hidden Markov model, lateral hidden Markov model, because they have the probability or emission probability of the amino acid type. Then I can use align each chain sequence against this hidden Markov model. So I build the final 3D structure of the protein complex. Now, I like to describe the details of this approach. First approach, how do you identify atoms and their amino acid types? So basically give a grade of the 3D density map containing the density values, only one single value for each vocal cell. I break them into patches, 3D patches. Then those are put as input for a transformer with attention layers, multi-head attention, then generate the higher level features. And actually we use the 12 blocks of these things stacked together. I take the output from the block 3, 6, and 9, then use them or the 12, use them to generate go through the convolution process to generate the higher level features. The output from these blocks are combined in the UNet fashion. It's very widely used technology for image processing. So basically the output from these 12 block are combined with this one, concatenated. The output concatenated is here, just in the UNet process, generates multi-level different features. Finally, they all concatenated here, use as input for the convolution layers to predict whether or not one vocal cell contains an atom, carbon alpha atom particularly, and the type, amino acid type, 20 different amino acid type is a classification problem. Now I get the classification results, carbon alpha atoms and their amino acid type predictions. Now this actually is naturally become a hidden Markov model because I can treat each predicted carbon alpha atom as a hidden state, right? So I can fully connect them because you don't know which one should be connected together. So emission probability for each hidden state actually is just the predicted probability of the amino acid type and considering the prior probability, background probability of amino acid frequency. So basically it's their normalized geometric mean of that to build the emission probability of 20 different amino acids generated from that carbon alpha state. Transition probability between 2 carbon alpha states actually is related to their distance between these 2 carbon alpha states. If their distance is close to the mean of the distance of contact residues or 2 adjacent residues in the protein you see in the data set, you got the highest probability. Otherwise, you get if you deviate from that mean distance, your probability decrease. So now we have a lateral hidden Markov model. Then we can use standard Viterbi algorithm to align each protein chain sequence against this hidden Markov model to build the entire structure, link them together to build the entire structure. It's customized Viterbi algorithm. So basically, you align first chain to it, you build one chain, first sequence. But because one carbon alpha only belongs to one chain, so we remove all those states, carbon alpha states that have been aligned. We get another smaller hidden Markov model. Then we align next chain to it, continue this process until all the chains can be aligned to that hidden Markov model. Now you build an atomic protein structure. Okay, now I describe how we train such a model and how we test it. So we use a data set called Cryo to struct data set we created. We publish in the scientific data. So you basically, here's the training data. It has 6,600 density maps. Here's a distribution of those density maps. Some of them are highly accurate, 1 to 2 angstroms. Most of them actually are 3 to 4 angstrom. We also have a validation data set that contains 740 density maps that are used to select the 3D transformer, which model works best. Then, after the model has been selected, we test them on the 2 standard data sets. 2 data sets. One is called a standard data set that has been used to benchmark the Phoenix, the most widely used method for initial method for constructing the density maps, protein structure from density maps. It has 128 density maps. Resolution ranges from 2.8 angstrom to 5.6 angstrom. It contains many residues, ranging from 400 residues also to about 80,000 residues. That's very large molecular machines. We also have a new test data set that has 500 density maps, that resolution 1.9 angstrom to 4 angstrom is a little more accurate than the standard data set because there are new density maps released to the ISMB. Also contain very large molecular machines. Now, let me see what are the results on these. So first is we evaluate the carbon alpha recognition results, how accurately our transformer can identify carbon alpha atoms correctly. We use 2 metrics. One is recall, percentage of correct carbon alpha atoms that have been identified. Another is the F1 score, basically consider both recall and precision, how accurate your prediction is. So you can see the Cryo-Struct has a 65% of recall, 25% percentage points higher than Phoenix. And the F1 score is also 14% percentage points higher than Phoenix. That's the first step. The second one is after we link those carbon alpha atoms into complex structure, protein structure models, then we compare those structure models against their true structures. Then we calculate what percentage of carbon alpha residues in your model can be aligned well with the true structures. So we call that a carbon alpha match score. It's widely used in this field. Our method, Cryo-Struct, is 53% higher than 42% of the Phoenix. However, this metric doesn't consider how big your model is, how complete your model is, only is a percentage of correctness. So if we consider the size of your model, because Cryo-Struct constructed much fuller, much complete model than the Phoenix. So we introduced a new score called the carbon alpha quantity. Basically, it's carbon alpha matching score times number of the residues in the structure models you build, divided by the total number of residues of this protein complex. So then you can see the score of carbon alpha quantity score of the Cryo-Struct is 0.43, almost double that of the Phoenix, because the more complete models have been constructed. Now, let's see how resolution affects the performance of these methods. We can see, on this data set, we plot the F1 score and the carbon alpha quantity score of the 2 methods against the resolution. So you can see blue line represents how the score change with resolution for Cryo-Struct. Red one is for Phoenix. You can see in both, as we expected, the accuracy actually decreased as the resolution getting worse, right? But the Cryo-Struct actually decreased slower than the Phoenix in this process. Then that's the standard data set. Used the density maps created years ago. Now we start using create a new large data set, not a density maps released recently, right, in the last year. So we have 500 density maps to rigorously validate the performance of these methods. So we also we plot the carbon alpha recall, F1 score, and also carbon alpha matching score, and also the carbon alpha quantity scores of the Cryo-Struct constructed for against the resolution. So you can see the quantity actually slightly decreasing as a resolution getting worse, and the average, the mean score over these 500 density maps for the recall is actually 70%. F1 score is 70% too, is higher than on the standard data set. The reason really is because you get a better data as input. So even the same method, you get a better output. Then for the model quality, after you build the atomic model, then also kappa alpha matching score is about 50%. Kappa alpha quality score is also about 50%. So it's also better than on the standard data set. That means if you get a better with data using the same method, you automatically get a better results. Now here, I'd like to show some examples, good examples today. We also have some failed examples too, but I'm going to show you the good examples. So here you can see one cryo density map. This is a very big density map. So that's a density map. That's a true structure. That structure we built. Actually, you have quality score 0.9, kappa alpha matching score is also 90%. It's a high quality. And the total number of residues we constructed is about 4,000 residues. Here's another reasonable examples. This is also a very large density map. It's about 2,500 residues. It's a true structure, reconstructed structure. Of course, there's some areas that have not been very accurately constructed because of the density information is missing in that area. So that's limited amount of information, structure information you can get from the density map are now. You actually need the structure models, co-evolution information to build that. When I submitted this abstract, it was not published, but unfortunately, recently, a couple of days ago, it has been published in Nature Communication. My student, Nabin, is a person who actually did this work. He spent 2 years of his PhD on this. Okay, finally got it published. He's very happy about it. We also deposited this source code in the bioarchive, including the prediction code, training code, data sets, everything. You can reproduce this work and to test how well it works. And also, I really encourage you guys to also work on this field. It's a surprisingly interesting field, but not well developed. Even they don't even have very good data sets. So that's why we created the Cryo-2Struct data set deposited in the Harvard Data Universe. It's a huge data set, terabytes of data. So that resource can be used by you guys to build your own machine learning methods for this problem. We also create a demo in the Code Ocean that you can actually, it's a live demo. You can run this program with the data to demonstrate how it works. So it's fully reproducible, this work. With that, I'd like to conclude my presentation with some key findings. The first is we developed a 3D transformer that produces backbone atoms from the density maps and amino acid types rather interestingly accurately, I would say, not perfect. Then we use subdensity maps, we use a innovative hidden Markov model to naturally align those sequences against the kappa alpha atoms we identified to build the 3D structures. By doing this, we think we have demonstrated that machine learning methods has great potential to automatically and accurately build protein structures from large density maps. Of course, this method can be further improved by using the predicted protein structures either as an input or as output in the post-processing step to further improve the quality of the predicted structures. With that, I'd like to thank my student Nabin for doing this work, and also my collaborator, Li Guowang, for providing critical advice for this project. Thank you very much.
Thank you for the nice talk. Congratulations to the student. I may work in Cryo-EM because of this now. We have a question there.
Very interesting. I've seen recently Cryo-EM work that is going the direction of trying to take snapshots of the entire cell. And then obviously what we will need is methods that will have to combine threading to guess from the shape, protein, the composition of the complex before you can even model it. But combining the two things.
Right.
So if you could expand a little bit on how far you are from being able to do that.
Okay, yeah, that's a great question. So Cryo-EM field is moving very fast in the experimental world, not necessarily in the computational world. So now, today I talk about how to use Cryo-EM to build large molecular machines, protein complex. Actually, use Cryo-EM, you can build the organelles, the cell structures, as Janet pointed out yesterday. How do you combine all these? That's called cryo-ET. electron tomography. They are related technologies. How do you combine this, integrate this, all this data to build the live dynamics of entire cells, including the molecular machines, proteins, and large organelles? Yeah, that's an open challenge right now, right there. We're trying to move in that direction. We're currently creating large molecular machines, but my collaborators always encourage me, hey, you need to work on the Cryo-ET. You need to transfer technology in the computational world, AI, AlphaFold, all those technologies to here to build that entire cell structure. That will be next breakthrough. Yeah, thank you for that great question.